Okay, so we keep this whole series as anatomy of questions, and here we want to discuss about the detailed structure of a question, because if you understand the structure of a question very clearly, you can answer them fast. Okay, and sometimes understanding the structure is more important, even important than knowledge of the subject itself. So today we will talk about unit 1 and unit 2 questions. Okay, so 6 questions from each unit. Approximate number of questions you can expect from these units in part C in CSIR NET exam. So this okay. So we are back with another session of anatomy of a question, and in this series now we are going to talk about unit 8, that is the genetics, human genetics. Mendelian genetics or non-Mendelian genetics, but mostly the question asked from Mendelian genetics are mathematical questions. So we'll solve that in the biomath classes, and we have already solved few of the mathematical questions in genetic classes already. So we'll mostly focus here on the non-math questions. We'll also solve few math questions, but non-math questions from genetics, and also the question from molecular genetics, particularly few chromosome reciprocal crossing over questions. Okay, so let's talk about that. Okay, so let's begin with it. Somatic recombination was caused by mild exposure to radiation on flies heterozygous for a given allele during specific stages of development, and the individuals were allowed to develop. Such individuals are likely to have. So this question is from somatic recombination. There are two types of recombination possible. One is the genetic recombination, another one is the somatic recombination. We all know about the genetic recombination. We have studied genetic recombination in details. We know the steps of the genetic recombination. But what about somatic recombination? In genetic recombination, the outcome can be transferred from generation after generation. While in somatic recombination, the process is done in the somatic cell. It's not in the embryonic cell. So in the somatic cell, if there is any sort of mutation, then when that cell divides into two daughter cell, all the daughter cell is going to receive that mutation. And a very common feature of the somatic recombination is that this mutation can spread from one part of the body to the other location of the body. So it's not tissue specific, but the genetic recombinations sometimes are tissue specific. Somatic recombinations are non-specific to tissue. Okay, so they can spread. Okay, this is one important property. Another thing is that's why, in individuals which are heterozygous, and if there is any somatic mutation is done, somatic recombination is done, then you can also observe the homologous feature in those heterozygous individuals as well. So the heterozygous individual having the somatic mutation and somatic recombination, you are going to see both the properties of heterozygous as well as homozygous present in that same individual after the somatic recombination. So these are the important characteristics of somatic recombination. So if you know this, then only you answer this, then this question is easy. But if you don't know, then you cannot answer it. So this is mostly a memory type. So clones of homozygous cell in heterozygous body, we can observe that. Yeah, the question is yes, it's true. Site specific mutagens will be produced? No. There is no site specificity, because the somatic recombination can give rise to the cells of the different locations of the body. So B is not true. C, twin spots, that is patches of mutant cells and homozygous wild type cells in the heterozygous body. As I told, this is a characteristics of a somatic recombination. So yes, we'll find that. We'll find both this type of characteristics of the homozygous characteristics as well as the heterozygous ones present. So A and C both are correct. D, tissue specific expression of the given allele, not possible. I told you this is not specific. So A and C are true. So option 3 is a correct one. So that's how easy it is once you know the properties of somatic recombination. But if you don't know, leave it. Let's go to the second type. This is a mathematical question, again a pedigree analysis. The inheritance pattern of a common trait which shows complete penetrance is shown below. Complete penetrance means 100% penetrance. That means every single individual shown in this picture which are affected carry the affected genotype, and those which are not affected shown does not carry any affected genotype. Okay, so that is already disclosed. It's nothing worry about, because all the Mendelian inheritance trait that we have discussed about are with complete penetrance. Now this problem gets complicated if they tell that this disease is following an incomplete penetrance. Then it will become much difficult to crack, but this is now easy to crack. Based on the average pedigree, fill the blanks from the opposite options given below. The trait is A. The probability that the child from marriage 3-1 and 3-2, that means this one and this two, means this is a child, either girl or boy. What is the probability of this child to be infected with the disease? And the first option A is what kind of disease it is, Y-linked or autosomal. So there is only two types of 50-50 chance, either Y-linked or autosomal disease. We need to figure it out. So let's do that. So first of all, you can clearly see that all the males are only affected, no females are affected. Now generally X-linked trait behaves like that, okay, but for an autosomal trait this is also a possibility. Remember, for a Y-linked trait, if this trait is Y-linked, then the Y is transferred from the father to the son. So all the son in the next generation must be affected, because all the son receives that Y from the father, right? So in this case, if it is Y-linked, then this individual, so third of the second generation must be affected, which is not the case. So this is definitely not Y-linked, this is autosomal. So we exclude option 1 and 2. Now we are only with option 3 and 4. So once you know this is autosomal, now whether it is a dominant or recessive trait, we see that this generation no one is affected, but this generation again someone is affected. So unaffected parents have affected individual, and this is a typical example of a recessive trait. So this is a recessive trait, okay. So for a recessive trait, so this individual is affected, so small a, let's say A is the disease causing gene, so small a, so the parents must be heterozygous or carriers, then only we get this individual affected. Now as the parents are carriers, this individual number 2 of the third generation there can have a genotype of capital A or capital A small a. We cannot tell which genotype this person will have for sure. And it is already told that the 3-1 is a carrier, so 3-1 is a heterozygous, capital S small a, this is fixed. So now we are dealing with this capital S small a, and this one we don't know, it can be capital A or capital S small a. Now we are trying to find out the genotype of this individual at the end here. So how to figure it out? Now think about it. Now the only way this individual will be affected, this new individual will be affected, if the parents are heterozygous. So what is the probability of this parent to be heterozygous? 100%, because he is already told. What is the probability that this mother is affected? 50-50 chance, half probability. So now if both parents are heterozygous, if both parents are carriers, then only this person will be affected. So if both parents are carriers, what is the probability of small a? 1 out of 4, isn't it? Because if you look at the Punnett square, capital A, capital A, small a, capital A, small a, small a, small a, 1 out of 4 is the chance of affected. So the chance of affected for this new individual is 1 out of 4 if both the parents are heterozygous. But we know that this parent, this father is heterozygous, 100% chance, but the chance of mother being heterozygous is half. So total probability of this individual to be affected will be 1 out of 8. So option 3 is with autosomal and 1 out of 8 probability. So that should be the correct one. Okay, this is how you solve it. Let's move to the next one. The following statements were made regarding chromosome pairing, shown in the figure below, and the subsequent segregation during meiosis 1 in the reciprocal translocation heterozygote. Now this is very interesting problems. Now they ask this kind of problems many times, okay. So they give you this kind of picture and they want you to explain the outcome, okay. Now whenever we are looking at this reciprocal translocation, actually there are two types of reciprocal translocation outcome. One type is adjacent type, adjacent cleavage, which is mentioned here in these statements also, we'll read that later on. And another one is the alternate type. So adjacent type and alternate type. Adjacent type means the segregation is done horizontally or vertically, okay, and alternate type means the segregation is done crisscross way, like that, okay. So where we are, okay, we are here. So I can show you this idea. You can see in this picture very clearly. So this is normal and this is translocate version. And if you can look, the adjacent segregation is always a segregation horizontally or vertically, but in case of alternate, the segregation is crisscross. So you will take this one with this one, so T1 with T2 and N1 with N2. But in this case, we are segregating T with N, T1 with N2, N1 with T2. Got it? So this is this type of question depends on the resolution after the recombination process is done. That means how exactly the chromosomes will be separated. Okay, this is very interesting. So you need to visualize the whole process. If you visualize the whole process in here, it says that the three ways of segregation in anaphase 1: adjacent 1 vertically, adjacent 2 horizontally, and the alternate. This is a truth. Yes, this is true, and that's why all the options carry this. So you cannot just answer it based on this option. Okay, every single thing is a truth. Second thing, the gametes resulting from adjacent 1 and adjacent 2 segregation will be non-viable due to the deletion. Now the question is, there are some properties. Again, you need to know the properties of this segregation products. Normally the adjacent products are non-viable. They generally don't give rise to any other gametes further one. Okay, so the gametes are non-viable, but the alternate product gametes are generally viable. So gametes resulting from the adjacent 1 and 2 will be non-viable because of the deletion and duplication. This is a true statement as well. So A and B both are true. If A and B both are true, then still we have three options left. Fourth option is gone. Now the third one, all gametes resulting from alternate segregation will be viable as they will carry both normal chromosomes as well as having the translocation in two poles respectively. This is a truth. As I told you, the alternate segregation products generally they become viable, so they remain viable. So that is also another important fact. So A, B, C all are correct, so option 2 is correct. I can answer that because I can know that, but I can explain this. If you look at here and if you check this horizontal segregation, will be A, B, C, D, E, F, G in one side, and on the other hand A, B, C, D, 5, 6, 7, 8, totally different. So the genes E, F, G is not present in there. Okay, so this segregation is not possible. For the alternate segregation, A, B, C, D, E, F, G and 1, 2, 3, 4, 5, 6, 7, 8. So for this segregation of this A, B, C, D, E, F, G, and on the other hand we have 1, 2, 3, 4, 5, 6, 7. Okay, but this one A, B, C, D, 5, 6, 7, 8, and on the other hand G, F, E, 4, 3, 2, 1. So we have this segregations as alternate. So it says that all gametes resulting from alternate segregation will be viable as they carry both normal chromosomes or both chromosomes having translocations in the two poles. So this is a complete normal chromosome, this is complete normal chromosome, or they both chromosomes having translocations in the two poles. So you see ABCD and the translocation in this pole, and again 1, 2, 3, 4 translocation in this pole. Okay, so this is a explanation for the alternate segregation. So alternate segregation always give rise to viable gametes, but the adjacent segregation will never give a viable gamete, because in all the type of adjacent segregation you will see some portion of like in this case you see ABCD, EFG and ABCD 5, 6, 7, 8. So few of them are matching, few of them are totally different, so it's not going to help them. Okay. Now this question, a co-transduction experiment was performed to decipher the linear order of 4 genes A, B, C, D. 3 sets of experiments were done where transductants were selected: set 1, set 2, set 3. Set 1 is for in respect to A gene, set 2 is for B gene, set 3 is for C gene mentioned in here. Screen for co-transduction and other markers. You need to find out the relative position of these genes. Co-transduction and the transduction frequency is used to map the genes present in the bacterial chromosome. And co-transduction idea states that genes that are placed very close to each other transduce together, they transfer together. Okay, so the more closely they are placed, the more the more frequency they have in the transduction. So the transduction frequency will be increased for them. So think about it. So the higher the transduction frequency, that means the closer the genes are placed. So utilizing this idea, we can still find out this map. How? Let's look at it. First look at the set 1. For this A with B, A with C, A with D. We can tell that A and D are placed very close, that's why the highest frequency. And A and C placed distantly. So we can write something like A and D closed, C distantly. Now in the set 2, we can see that B is most closely placed with C and B with A is distantly placed. Okay, so B is very closely placed with C and B is distantly placed with A. This is quite justified with this. Okay, so based on this idea we also put B in this case. Third one, between C and A no frequency. That means definitely C and A are placed in opposite direction, the most distant location. So that means this is not true. A is present in one side, C is present on the opposite side. Now either B and C or like this. So as per this, what we can see, C and B more closely placed than C and D. Okay, so now the idea comes here. We know that A and D is very closely placed, so A and D. Then we also know that the B and C very closely placed. Okay, so B and C closely placed. There is a distance between B and D, and B and A is also distantly placed. Okay, so that means from there we can get this idea A, D, B, C. This should be the option. Okay, so now if you look at the options A, D, B, C, that's option 4. That should be the correct option. If I show you, this is how you can find it out, A, D, B, C. And you know that A and C are most distantly placed due to the least frequency for them. Then CB, CB is also very distantly placed. In this case CB 69, but in this case also CB 78 distantly, and DB 68. You can see the B with D 68 and A with B 31, let's say, in that case. Okay, so this is how you can put the map together, okay, to find out. So they don't ask you like the distance between each of them, just I put it here, but just need to know the position, because this co-transduction frequency is not enough to find out the distance between the genes, but we can find out the position, relative position of the genes quite well with the help of this. Okay, so A, B, D, A, D, B, C, that should be the answer. So let's move on. Sickle cell anemia is a recessive genetic disease caused due to the point mutation in the 6th codon, abolishing one of the MSP2 endonuclease digestion site present in the beta globin gene. MSP2 digest DNA from a normal person gives two bands, 1150 BP and 200 BP base pair. In the beta globin gene, we get two bands, okay, normally for a healthy individual, and because there is this 6th codon, and if we treat it with MSP2, it cleaves from that point, okay, from that point of the mutation. In beta globin gene, a family with a proband based on the disease phenotype give the following MSP2 digestion pattern. So what you can understand reading this question can be in unit 13, can be unit 7, can be unit 8, any place. Okay, so we know one thing, that for a normal healthy individual, MSP2 can cut. So healthy individual, MSP2 can cut into two fragments, 1150, 200. For a diseased individual, there is a mutation, and due to the mutation, MSP2 failed to cut the DNA. So one band only, and then band of 1350. Okay, so for a diseased individual there will be one band of 1350. For a healthy individual, it's 1150 and 200 BP together, two bands. So if you look at this thing, we have this the leader, and we have this ladder sequences with 1350, 1150, 200. We find out that the mother has two bands, like 1150 and 200. That means mother is normal, father is diseased. Son 1 is normal because son 1 only have one band. Daughter, in this case, diseased. You can think daughter also have 3 bands. Okay, son 2 also diseased. Think about it. Daughter is not diseased because daughter also have 1350 bands. So it's a possibility that daughter can be a carrier here. Okay, so what you can tell from this idea, the father is diseased obviously, son 2 is also diseased. Anyway, son 1 is proband means diseased, and the given mutation is not present in son 2. Yes, it's a true statement. A is true. If A is true, then option 3 and 4 are wrong. Okay, the daughter is a carrier for the given mutation. Let's see. The gene is X-linked, thus SUN1 becomes the proband. It's SUN1 is not a proband, so C is not correct. SUN1 is normal. So C is wrong, so option 1 is left. You don't need to read any other option. You don't need to read any other statement, you know the answer. But let's read it. The father and daughter are affected. Father is affected, daughter is affected. We don't know whether the daughter is affected because the daughter also have 1350 band. So daughter is not affected. D is not true. A de novo mutation in the same site of normal allele has allowed appearance of this disease phenotype in the proband. So whatever it is, it's true or false, we know it's true because it's a part of this option. De novo mutation in the same site of the normal allele has allowed the appearance of disease phenotype in the proband. Yes, there's a possibility. Now the thing is, the daughter is a carrier and that daughter can transfer this to the next generation. Mother is normal, father provides the X. This is X linked. One of the X. So the trait is something like this. The father contains the disease. So this is a healthy mother, and one of the the mother is a carrier. It may be a possibility that mother is a carrier, because if that's the case, then one son can be affected, the other son can live healthy. But it's not also a possibility because mother is totally healthy. It's not about the carrier even. So you don't know that there's a second mutation that is done somewhere to make this normal allele into a proband. Okay. So this is not even a normal way of understanding the pedigree. But we know one thing, the statement 1 is true, and we also know statement B is also true. The daughter is a carrier at the end, because the carrier should carry this extra 1150 and 200 along with the 1350. Okay, so this is how you can answer that. Now this one, in transgenic mice, the orientation and location of LOX-P sites determine whether the Cre-recombinase induces a deletion, an inversion or chromosomal translocation. If researchers want to put the LOX-P site in such a manner that only inversion will take place, which one of the following construct best justifies their intention? And the gene X is the target gene that we are seeing. The answer is already mentioned. I should not have mentioned that in there. Anyway, the thing is that you cannot answer this question if you don't know the outcome of a recombination. Just like the translocation at the end, we talked about one problem earlier, and this is also a Cre-lox recombination. And it's not about Cre-lox recombination, it's about any kind of site specific recombination where it depends on how the LOX site is placed, based on which the inserted segment can be deleted. The segment in the middle, actually the gene in the middle can be deleted, can be inverted, okay, or can be translocated. So there are this option, if we want this gene, this X gene to be deleted, or so we call it the X gene to be inverted. You see the manner that only inversion will take place for the gene X. So obviously the LOX-P site should be present in both the sides of the gene X. So this option 1 is not right because the LOX-P sites are present totally different location. 4 is also not right because again present in totally different location. So obviously some point between option 2 and 3. Both the cases the LOX-P site is present in the opposite side of the gene X. Now in one case, in the option 2, the LOX-P sites are in the same direction. In option 3, the LOX-P site in the opposite direction. So if you know this mechanism, normally what happens, if we put the inverted repeat, then it causes the deletion, and if we put the same direction, that causes the deletion. Okay, so if we put the LOX-P site in the same direction as a direct repeat, they cause the recombination in the second time, and that causes the deletion of the gene. We don't want deletion, we want inversion. So obviously we need to follow the inverted repeat. So we need to put the LOX-P in the inverted structures surrounding the gene X, so that it can cause the inversion of the gene. That means earlier there is a blue side and green, now it will be green, blue, inversion. So this inversion is 180 degree inversion. Okay, so which one should be correct? Option 3. Okay, that is the inverted repeats. Always remember, inverted repeats cause inversion and direct repeat cause deletion. Inverted repeat causes inversion. That's clear. Another question, just like the translocation process, reciprocal translocation. The following diagram shows meiotic pairing in an inversion heterozygote and a point where single crossing over has occurred. So this is a point, single crossover between a single strand of the chromosome here we are seeing, okay, and the other is remain intact, okay. So the problem in this question is that the 3D visualization, you need to visualize this outcome, the 3D visualization in your brain to answer that. Sometimes it's not getting clear, that's why people get it difficult to answer in most of the cases. So think about it. It states that the resulting gametes produced may have the chromosome having normal gene sequence, the chromosome having inverted gene sequence, a dicentric chromosome with duplication and deletion, an acentric chromosome having duplication and deletion, the chromosome having duplication and deletion. Now this is, there are so many options are given. One thing, if this is the way to segregate, then we have one A, B, let's write it, A, B, C, D, E, F, G. Nothing happened in that side. This side, again A, B, C, D, E, F, E, F. So this is A, B, C, D, E. So there will be A, B, C. This one will be A, B, C. This is D, and then afterwards it will get, because this is what it will get, another AB, CAB, something like that, okay. So this is gene duplication, AB, AB, C, these are duplication, and deletion of EFG in this strand, this is fine. So this is normal, and in the other chromosome we'll see a deletion of EFG and duplication of ABC. The same thing happens for the other one as well. So which statements are correct? The chromosome having normal gene sequence, yes, few of them will have normal sequence, actually among the gametes, because there will be 4 gametes, one gamete will have this, 1 or 2 of them will have this normal. The chromosome having inverted gene sequences, yes, will also be there, and the chromosome having duplication and deletion, E, is also true. A dicentric chromosome means a chromosome with 2 centromere, and acentric means chromosome without any centromere. Both C and D are not true, because the crossing over is taken place right in a downstream side of the centromere. Centromere is kept alive, there is no segregation taking of the centromere from there, okay. So as a result, the centromere will remain as it is, only the fragment of gene is swapped. So there is no way the dicentric or acentric chromosomes will be produced, although the duplication and deletion will take place, but no dicentric or acentric chromosome. So C and D are wrong. So any option with C and D are wrong. So all the 3 options 1, 3 and 4 contain C and D. So the only option left is option 2 that carries statement A, B and E. All the 3 statements are true. So this option 2 is the correct one for this question. Okay. So remember what I draw here is, when I see this question, you need to draw the simplified version of this gene, and when you draw the simplified version of this chromosome, you can easily answer these questions. So visualize that in your brain and write it in the paper. Do the rough work and you can go close to answer this kind of questions. Okay. Yes, the answer is yes. If you ask yourself this question, that can you qualify CSIR NET Life Science exam within 6-month preparation? Yes. And if you don't need to believe me, just see the results that our students are getting who joined Shomu's biology coaching for 6-month period of time.
So I'm really thankful for Shomu's biology, and I would suggest everyone to go for it.
A lot of help and support by Suman sir, always remembered in my mind.
The study materials I got helped me to understanding the topic very well, and the test series helped me to covering up the syllabus in a perfect time.
This is all because of the online test series. in depth knowledge what Shomo's Biology gave me. I want to thank Shomo Biology. I want to thank each and every member of Shomo's Biology for their effort, whatever they are putting on us.
And I am very much happy to choose Shomu sir for my CSIR NET JRF journey.
Really leveled up my preparation. I was confident on the day of exam.
The classes helped me a lot to understand my subject from very basics to advanced level.
So I really thanks Shomo sir and also ma'am for always guiding me.
With proper guidance, roadmap, and the help and training with perfect amount of study material and how to utilize them, the resources, everything together can help you qualify that. I am the person who will help you reach to the pinnacle of your success and your journey for the CSIR NET Life Science examination. If you trust me, if you trust my lectures, then join Shomu's Biology online coaching. Registration is going on. The details are there in the description. Fill the form to reach out to us or simply contact the number given in this video.